Good morning, everybody. How are you? Okay. Well, as Miss Putney or Miss Mendenhall said, my name is Pamela Wade, and I'm here from the Monterey Bay Aquarium this morning. I'm so glad to see you all. And I'm going to be talking about my research in the Elkhorn Slough, and I have some pictures to show you. So let's get started. I'm going to stand right here. All right. So my background is that I like to study invertebrates. And I'm curious, who knows what an invertebrate is? What does that mean? Oh, I don't see any hands. Maybe turn to somebody next to you and share what you think an invertebrate is. Go ahead. Okay, it sounded really quiet. Yeah, okay, cool. All right, let's see. Show of hands. Who has an idea what an invertebrate is? I see a hand over here. What about this table in the middle over here? I saw some hands. Oh, okay. Who has an idea what an invertebrate is? I heard lots of conversation. Well, I'll give you a hint. Yes. Exactly, an animal without a backbone. And if you look up here on the screen, you can see some examples of animals without backbones. Down here in the right-hand corner, there is a sea anemone. That's the orange, reddish-colored animal. There is a sea star up in the top left-hand corner. So these are examples of invertebrates. The animals that I study are these in the picture. They're marine invertebrates, so they all live in the ocean or in the surrounding habitats. Okay? Now, today I'm going to tell you about my research that I did for my master's degree. And it took place here in the Elkhorn Slough. So how many of you have driven down Highway 1 and you've passed the power plant right by the ocean in Elkhorn Slough? Has anyone seen that before? Or maybe some of you have gone hiking with your class at Elkhorn Slough or gone kayaking? Well, that's where my research took place. Now, this is a really important place because the Elkhorn Slough leads into the Monterey Bay. And if you take a look here, at the bottom of the picture, there is the opening to the ocean, and that's where there's a break in the sand, and the water from the slough goes right into the bay. Now, if you were to travel way up the slough 7 miles to the end of the slough is where my research took place. Now, why it's important that there's an opening is because this was man-made. So people made this opening. The Salinas River changed its path over time, and now the water that goes in and out every day with the tides, that water carries a lot of the sediment, the sand and the mud from the slough, in and out. And when that's carried out, it causes a lot of erosion. Is everybody familiar with the word erosion? Yes? Okay, it causes a lot of erosion, and this is important for what I'm going to tell you later on. So the animals that I studied are these. They're Olympia oysters, and they're a really small oyster. They're only about 5 or 6 centimeters, so about as far apart as my fingers are, as I'm showing you. And these little Olympia oysters make their home in the Elkhorn Slough. They're native to California. They're actually the only oyster that's native or from California. And they used to live all the way from Alaska down to Baja. But now their populations are really small. So we're concerned about the oysters, and that's one reason I studied them. And my focus is marine ecology. So I'm interested in not only the animals, but how the animals survive in their habitats. So here's another picture of the Olympia oysters. And what I want you to notice is they live in communities. So Olympia oysters live together. You can see how they're all kind of clumped together. And they make up an oyster reef, which is really important because this reef creates a home or a habitat for other things. So lots of other little animals would live among these oysters. The oysters also filter a lot of seawater. So they take in the water, they are taking the plankton and the bacteria out of the water, and then they release the clean water back out. So that's really important. But these oysters do not do well if there's a lot of sediment. So if there's a lot of sand and mud covering them, are they able to filter feed? No, not at all, right? They can no longer get their food. So that's something that we need to consider in the slough. Okay? These oysters are also important food for other animals. So lots of animals in the slough like to eat them. So if their populations are low, then the other animals might not do so well either. So what's happening in the slough is there are a lot of animals being introduced that aren't from the slough originally. And the one on the left, this crab, is called European green crab. It's been introduced to the slough, and it likes to eat a lot of the other animals. It would eat oysters, it would eat other invertebrates that it can find, and then it lowers their populations. Now on the right-hand side of the screen, these are Batellaria, or little mud snails. These little mud snails love to eat the small plankton that's just on top of the mud, but they can grow really fast, and there are a lot of them. And they take away habitat from other animals in the slough. So you may be wondering, how did these animals get here? Do you have any ideas? Where did these animals come from? Yes?
From the water?
Oh, yes, from the water, you're right. How did they get here from the water? How was the water carried to the Elkhorn Slough? I'm giving you a hint on the screen.
Boat?
Yes?
Shipment?
Yes, by ships, that's right, and shipments. So what happens as boats travel, animals can hold on to the side, or we call it the hull of the boat, and they attach because they need something to hold on to. Now, animals can also travel in the water, as you mentioned, inside of boats. So big ships pump water in, and that helps to stabilize them as they're traveling, and then they pump the water out when they get to a new place. By doing this, they release a lot of little plankton with them. So that's one way that animals can get transferred from one place to another. And that's how a lot of animals have been brought here to the Monterey Bay. Additionally, there used to be oysters raised in the Elkhorn Slough. So the picture that you see on the right is a man-made oyster reef where they're raising oysters. We brought oysters from Japan and from the East Coast, and we raised them in the slough up until the 1980s. So for quite a long time, over 60 years, from about the 20s to the 80s. Well, along with these oysters came lots of other animals. So we introduced other animals like those mud snails accidentally. So now that there are all these, we call them non-native animals in the slough, they're taking away some of the space where the oysters would live. And what you see in this picture is tube worms surrounding, look very closely in the middle of the screen, and those are the oysters. Now, you may also notice, besides the tube worms, these non-native species, there's a lot of mud around the oysters. Now, is that good for them? No, not at all, right? They're getting covered with mud. These oysters are sessile, which means they're stuck to a hard surface for their whole life. So wherever they land is where they're going to grow, and they can't move from that place. So they don't want to be covered by mud. So here, you can see on the left-hand side of the screen, this is a tube worm reef. Now, these tube worms have a really fancy name. They're from Australia, and their scientific name is Phycopomatus. Huge name, but what it means is that these are tube worms that have a hard skeleton or tube, and as they reproduce, the new tube worms land on the adult tubes, and they keep growing. So they can form reefs bigger than this table, or longer than this table. Okay? So they are throughout the Elkhorn Slough, and if you look up close, this is what they look like, just tubes on top of tubes. So they also make a lot of habitat where other animals can live. But what scientists have found is the animals that live among their tubes are usually other non-native species. So these tube worms are taking up a lot of space in the slough, and they like to live in the areas where the oysters would normally live. Now they are also sessile, which means they live on this hard substrate, they don't move, and they filter feed, so they're eating the same food as the oysters. So from my research, I spent a lot of time in the slough, and I'm going to show you some pictures of what that looked like. But I asked some questions, and I don't think they're going to show up today, but I'll tell you what those questions were. Okay? So the first question I asked was, how do all these non-native species that are introduced affect the animals that are already living there? So how do they affect those native species? The second question I asked was, does the water level, so the height within the water, does that matter? Would animals, at a higher level do better, or those at a lower level? Okay. And then I asked a third question, and the third question was, do the adult oysters have a better chance of surviving than the babies? I thought maybe it's harder because the babies are smaller. Maybe they get overgrown by these non-native species first. So these were the things that I set out to investigate. And I started my investigation in May of 2014, so a few years ago, and I spent almost 2 years investigating and then writing up this project in order to get my master's degree. So what you see here is a little light, but there's a picture of the Elkhorn Slough. Does anyone remember how long I said it was? Say it out loud. 7 miles long. So my study site, the place where I did my investigation, was all the way up at the top, the furthest from the Monterey Bay. Okay? And there's a little red circle to show you where that was. This is what my site looked like. So there was a lot of water, there was a lot of mud, there was a bridge that ran through the site, and closest to us in the front, these are culverts. What that means is these are pipes that the water can go through to travel to a pool behind the site. That's something that people have made to help control the amount of water in this area. So with all these new hard substrates like the culvert and the bridge, those are places that animals could settle that they wouldn't normally. Okay? They would normally settle on natural surfaces like rocks. And down here in the bottom, the picture is of one of the pilings of the bridge, so one of the poles, and it's a combination of tube worms and oysters. So I chose this site, which is called Hudson's Landing, because it had both the non-native species and the native oysters living there. So I thought this was a good place to go study oysters. All right. So this site, as I said, was full of mud, very, very muddy. You had to wear a wetsuit, like my students that went out with me here on the left, this is Meg, or hip waders, like I was wearing on the right, in order to get into the mud. And it was really hard to walk around. So as you're moving, sometimes your knees are all the way in the mud, sometimes even deeper, and this mud is full of water. So remember, the tide's coming in and out, so you can only be out there when the tide is low. When the tide is high, it would have been over my head. Yes?
Have you found any animals in the tide?
Have I found any animals in the water?
Yeah.
Yes, I have. Great question, and I'm going to show you those. Okay, you get to see what they are. So thinking about where you do your studies, it matters time of day. It matters if you're working in the ocean or in an estuary like this one. If the water is going in and out, you have to consider all these factors. So what I did first, to answer your question, is when I first started my study, I needed to learn what lived there already before I put any experiments out. So I took out an intern, this is one of my students, and she and I laid this transect line, which is a long measuring tape that we laid throughout the site, and we sampled a number of places to figure out what lived there. So we couldn't sample the whole thing, that would have taken us a very long time, but we sampled along this tape at 2 different heights. So we sampled at 1 foot above the average low water, and we sampled at 1 foot below. Okay? So we wanted to cover that whole area that we call the intertidal. And the way we sampled this was to use a quadrat. This is a scientific tool. It's super simple, but what you do is you lay this quadrat or this square over the area that you're studying, and then every place where the ropes cross, that's a point, and you sample what's under that point. And we recorded that on a piece of paper. Okay? And this gave us an idea of what animals live there. So I put up some pie charts for you to see, and I'll explain what this means. But what it's saying, and this was at the lower tidal height, we're going to start with the lowest one, is that in the green at the top, the 41%, was tube worms. So a little bit less than half of the space was taken up by tube worms. The orange, that little piece of the pie, which was about 3%, was oysters. So very few oysters. The yellow is a type of sponge, which is also an animal. And then the purple is a barnacle, which is an animal with a hard shell. And the bottom, the 47%, is bare space. That means nothing was there, it was just mud. Okay? And we did this again looking at the higher tidal heights. So we started with the low one, looked at the high, and we noticed, again, there was a lot of tube worms. It was over half of the space was taken up by these tube worms. There was a little bit of sponge, and then about 26% was bare space. So this is what we found out there naturally. And then what I did to set up my experiment was I knew I needed to put oysters out into the slough. There are very few oysters there right now. The population is pretty low, so I needed to outplant them or put them out. Now, I had two experiments. The first was on adults. So I wanted to look at those adult oysters because I was asking that question, which one's going to be influenced more, right, by non-natives, adults or the juveniles. So on the left-hand side of the screen, what you're looking at is a settlement plate. Now this is a little square plate that was about 10 centimeters by 10 centimeters, so smaller than half a sheet of paper, and there's one adult oyster on there. So every plate that I put out had one oyster. I put them at those two different heights within the water, and I'll show you how I did that. And then the plate on the right is covered with 20 little baby oysters, and that's the second experiment that I'll talk about in a few minutes. So we'll talk about the adults first. So here I am out in the slough, again I'm in those waders, and what I'm holding is oysters. So I needed to start by going out and collecting them, because I didn't have any at this point. So I went out to a place that's near my study site, but not exactly. And the reason I collected oysters from the slough is I didn't want to introduce any other new species. So I wanted to collect from the site that I was going to put them back into. So I'm holding some oysters here. They're attached to rocks. And here's a close-up of the oyster attached to a rock. Do you notice anything else in that picture? What else is attached to the rock? I heard it. Say it louder. The tube worms, right? So even the oysters that I was finding attached to rocks had tube worms on them. So I needed to take these back to the laboratory, and the first thing I needed to do was clean off anything else living on those oysters. So I scrubbed them with a wire brush, and I scraped them, and I soaked them in fresh water to make sure they were completely clean. It doesn't hurt them at all because they have a really tough shell. And then next what I did was I have all these cleaned oysters. I needed to attach them to the settlement plates. And what I was trying to do is standardize, meaning make everything the same that I was going to put out. That way I wouldn't have any added factors in my experiment. So I took each of these oysters and I used this really cool marine glue that you can actually glue things underwater by mixing this together. And I glued each of these oysters to a plate. And that took quite a while. So I glued them all on, and then they had to sit underwater for the glue to harden. And then what I did next was I gathered all my supplies. Okay, I gathered up all the oysters on their settlement plates. I built these racks, which is just PVC pipe, but they were very tall, about 5 feet tall. And I drilled holes in them, and then I added the plates, and I stuck these out in the slough at that site that I showed you at Hudson's Landing. 2 different heights, so 1 foot above, 1 foot below. And then I left them alone for 2 weeks. Because remember, everything's been cleaned off. Now I need those non-native animals to settle. So when I came back 2 weeks later, what I found was on my settlement plates, you can see the one on the left, the tube worms had started to settle. And then I went back every 2 weeks from this point on for an entire year, and what I did was half the plates, I left the tube worms and the oyster, but cleaned off anything else that was there. So I used a brush, I used tweezers, I had to clean off all the other animals. And then the second plate, I left the oyster but cleaned everything entirely. Because I was trying to see which one would grow more. Would the oysters by themselves grow? Or would the oysters with the tube worms grow? Or would there be competition? Okay. And then I had that second experiment with the adults where down in the bottom half of the screen, there are oysters with other non-native species, so no more tube worms, but things like tunicates, which is another animal, and there were bryozoans, which is another animal that grows all over the plate. And then next to it on the right, again, I cleaned everything off but the oyster. So any ideas? Which one do you think grew more? The oysters by themselves or the oysters with other animals? Okay, here's some ideas. How about if you think it was oysters by themselves, give me a thumbs up. If you think oysters with other animals grew more, give me a thumbs down. Okay, I see some different ideas. Well, we'll talk about it. I'll tell you what I found out. So as I said, I went back every 2 weeks. I always had help, usually from either another graduate student or one of my students that I teach. And we went out every time was low tide, and we had to take every single settlement plate off of these racks. We had to measure every single oyster, and we had to take a photograph of every single plate. And sometimes this was 4 in the morning. Sometimes this was 8 o'clock at night. It just was whenever that low tide was. I didn't get to choose. Okay? And I would stand in the water, and I did this for a whole year. And I wrote down all this information that we recorded, and then when I came back to the lab or the aquarium, I used the pictures that we took, and I used the computer program to measure the oysters in a different way. So when I was in the field, I was using something called calipers, where you can actually measure length and width of the animal. In the lab, I used a computer program where you can measure area. So I wanted to figure out if the area, if I did the math, was the area really the same as the length and width, or would it be a little bit different? I found that the area was a little bit more accurate. I also used a second computer program where you lay a quadrat over the plate. And does everyone remember that picture of that quadrat, that white square that was out in the field that I laid down? Yeah? Well, this one is a little tiny picture of one, but it has 100 points. And I had to sit there with my computer and figure out what's under each of these points, what different non-native species. And that's where I found out what lives there. Okay? So I did that, and then from that information, I made a lot of graphs. So I graphed the information to be able to share it with others and share it with other researchers. So what you see here on the left is that lower tidal height, which is 1 foot below the average water, which in meters is -0.3. And then on the right is the higher water height. And what the little dots are on the screen is when the tube worm was there on the tile or on the plate, that's the green dots. When there was no tube worm and it was just the oyster, those are the yellow dots. So looking at these two graphs side by side, do they look exactly the same? No. Can anyone tell me what looks different? What looks different?
You can talk about color. What color?
Sure. What color do you see on the picture on the left? What colors? Want to describe it?
But there's mostly orange.
Okay, so you see more of the orange. Which dots are on top on the left, the green or the orange? The green, right? Which dots are on the top on the right? The orange. So what it's saying is that on the lower in the water, we saw more oysters growing when there was this competitor, this tube worm, which is really strange. This is not what I expected to see. Okay? On the right, we're saying when they're higher in the water column, we were seeing that when the tube worm was removed, they grew more. So that's really interesting. So I had to figure out why that was. Okay? All right. So we have all these graphs, and as I went on with my work, I had to figure out why this was happening. Let's see if it'll go to the next slide. Maybe. I'm just going to give the computer a second to think about it. Think about any questions you might have about what we've said so far. Thinking. Does it look fun to be out there in the mud and the water? Yeah, I thought it was a lot of fun. It's still thinking. Yeah. Okay, awesome. Yeah. Awesome, thank you. All right. So the second picture, I'll show you this one real quick, is the same as the first, except instead of tube worms, this is when other species, other non-native animals were there. And we saw similar results. Actually, at the lower tidal height, they were very different. We saw a difference in the growth of the oysters. When the non-natives were there, we saw the oysters grow a lot. When there weren't other species, they didn't grow much at all. And then at the higher tidal height, it was very similar. So again, this was really confusing to me. I wasn't sure why this happened exactly. Might have to do it the other way. Yeah. Yeah, let me see. Okay, so I made some more graphs to explain what I saw, and what I noticed was the animals that settled on the tiles, those non-native species as we're calling them, look very different than that initial survey I took. Okay, so what I found that we were finding different animals. We were finding animals like the tube worms, okay, which we saw originally. We were also finding things like brown algae, which isn't an animal at all, but we saw a lot of brown algae. We saw a bryozoan, which is a funny animal that looks similar to snake skin. It kind of lays across the settlement plate. And then we found these mussels, which you can see down in the bottom right. And then we found this really funny animal called a tunicate. It has two siphons, so it takes water in and out of its body, and sometimes people call them sea squirts because they have soft bodies. Well, what I didn't know was what species some of these were. So I didn't know exactly what they were. I had an idea. So what I did next was I went back to the lab and I brought some of these animals with me because you need to be able to identify them correctly to know if they're actually non-native or not. And I ran the genetics. So I extracted DNA and looked at that to find out what these animals were. And I actually found out that some of them were pretty rare and others were very common, but they were non-native. So that was really interesting to me. And then I decided it's time to set out that second experiment, right? Because we set out the first, we looked at the adults, we got some really strange results that I didn't understand, but then I needed to set out the juvenile experiment. So it's a little dark, but what you can see on the left is a tank. This is back at the Monterey Bay Aquarium. And on the right, there's a second tank. The first tank is filled with oysters. The one that's all dark is filled with oysters and algae. The tank on the right that looks like it's clear is just salt water. But I needed to heat up the salt water because in order to have baby oysters to sit out in the flue, guess what I needed to do? Any guesses? Where do we get baby oysters? I heard some ideas. What was it? Yeah, from the adults, right? I needed to cause these oysters to spawn or reproduce in order to get babies. So I did this in the lab, and this is really cool. If you know anything about oysters, I didn't know much about oysters before I started this, but oysters are typically both male and female, okay? The oysters that I worked with are really neat because the female oyster actually raises the babies inside her shell until they're old enough to swim on their own. So I thought that was really cool. So what you see on the left is the adult oysters. Adult oysters mean an oyster is over a year old. The males release sperm into the water, and the female takes this into her shell to fertilize the egg. Okay? This stays inside her shell, this fertilized egg, until it starts swimming. Now, earlier I told you that oysters stay sitting, right? They are sessile. These baby oysters are actually able to swim. So what you see in the top right, where it says Petitvelliger, that's a little baby swimming oyster. They settle out, which means they land, and they begin to grow. And then, this is the crazy part. So this oyster at a year old is first going to be male, and then guess what? You knew, it's going to turn into a female. So every time they reproduce, they switch male, female, back and forth their whole life. So this was hard for me to know if I had males or females, because you can't tell by looking at them. So I needed to have a lot of oysters that I collected to put in the tanks to make sure I had some of all different ages, right? So that I had some males and some females. So I put about 25 oysters in each of my tanks. I had 4 tanks total, and I raised the temperature. I fed them a lot of food. Any ideas what they eat? They eat plankton, that's right. So I fed them plankton that we raise at the aquarium. We raise plankton in big tubes, fed them that 5 times a week. And then when they spawned, I used a flashlight to look through the side of the tank, and you could see all the babies swimming around. It was so cool. So then I had to take the babies out so the adults wouldn't accidentally eat them. And I put this in, and I made this. It's just plastic mesh with these settlement plates attached to it. Put this inside the tanks, and then when the babies were ready, you can see down here in the right-hand corner, they would land on the plate, but they're so tiny, they're smaller than the tip of your pencil at this point. They land on the plates and they begin to grow. And then what I had to do was the same thing as the adults, is outplant them or put them out into the slough. Now, the pictures here, if you can see, it's a settlement plate covered with baby oysters. So the difference between the babies and the adults is they grew so fast. I standardized the plates, which means I removed oysters so there were only 20 per plate when I put them out into the slough. And I did the same experiments as the adults, looking at the other non-natives with the babies. And what I found was that these little tiny juvenile oysters grew so quickly, I couldn't even leave them out there a year because they covered the entire plate. So any guesses what this tool is that I'm using to measure? I mentioned it earlier. Starts with a C. These are the calipers, and this is a close-up picture of the calipers I'm using to measure the length and width of the juvenile or baby oysters. And then this is my favorite picture. I just wanted to share with you because it shows from the time I put them out, within about 6 months, how they went from little teeny tiny babies, which is about the size of a pencil eraser up here, each one on the top left, and then down to the bottom right is how big they grew within 6 months. And I did the same analysis as I did with the adults, is looking at which grew better, if they were with or without the other non-native species, in this case the tube worms. And what I found was that with the juveniles, it really didn't matter. It didn't have the same effect as it did on the adults. So this was really interesting to me. All right. So then I asked myself, why? So why, if it mattered for the adults, didn't it matter for the juveniles? We're going to see. We're going to get this to go back in. Okay. So again, these would be the questions, but they're not showing up. So the questions were, do these non-native species affect the oysters? And was it different for adults versus juveniles? Also, at which tidal height did it matter more? So I answered the questions, and I had the help of over 20 different folks, from other students, these are students of mine, to folks like your teacher, Ms. Mendenhall, helped me out in the slough a lot, other graduate students. It takes a lot of people to do a project like this. And what I did was then present all the data that I had to answer these questions. And I shared it with the folks that work at the Elkhorn Slough in order to help them better take care of the animals that live there. And what I found out was that oysters that are outplanted, that are about the size of a pencil eraser, are going to have a better chance of surviving around these non-native species than oysters that would settle out there on their own. And what's come out of this is pretty interesting. Actually, the folks at Elkhorn Slough that run the estuary reserve are thinking and trying right now to raise juvenile oysters in the lab to outplant in the slough because they want to increase the populations. So I've been contacted about that. So it's pretty cool to find out that there is research going on now because of a project that I started a couple years ago. And this is just a fun picture of all the people that helped me and all the fun that we have to have out in the field. And I would encourage you, if you like science, to think about projects like this, especially as you move on through school and go to college, because there are so many fun projects. And I just want to point out that I designed this myself, and I built all the materials myself. So you didn't need a lot of money to do this. I made everything in my garage, which was really fun because I got to play with tools too. Do you have a question? We did find ducks. Everyone likes to ask about that one. So down here, standing on the boogie board, because I was using a boogie board kind of like a table out there to hold my materials, we found some baby ducks one day that I don't know what happened to the mom, but the babies were left alone and they swam up to us, and we had to stop the research and take them to the SPCA to be cared for and then go back to the research. So it was a lot of fun. Any other questions? Oh, some hands. Yes. Am I famous? Not really. No. But thanks for asking. No. I do get an opportunity now to present this project to other schools, and I get to go to conferences, but I'm not really famous. Yes.
Will you work at the Monterey Aquarium or wherever you work until you're retired?
Yeah, I think so. So the question was, will I continue to work at the Monterey Bay Aquarium? Yes, I've been working there for 14 years. So I worked there before I started graduate school, and I still work there. And I'll probably be there a long time. If you get to visit with your school, you may get to see me. I teach a lot of the programs there, and I run a middle school program there too. And Ms. Mendenhall will tell you more about that. Any other questions? Yes. Say it really loud. I can. How does this affect you? Good question. Well, you go to school and you probably live in Salinas, right? And even though we don't live right on the Monterey Bay, we are all affected by the bay, right? We rely on the Monterey Bay, and we affect the bay too. So can anyone think of a way that our actions here affect the Monterey Bay? What do you think? Is there anything that we can do for either good or bad to affect the bay? Yes. Stop littering? Yeah, definitely. If we litter here, even though it feels like we're far from the bay, it gets carried by rivers and streams and by the wind into the bay. What else? Say it really loud for me.
Not pollute the water.
Absolutely, not polluting the water. Anything that's on our streets that goes into our storm drains will end up in the bay. Something that's really important about the Elkhorn Slough, it's a wetland. It's also an estuary, which means that there's fresh and salt water in this place, but it acts as a big filter. So not only are these oysters a filter, but that surrounding land is a filter. So when we have things that are on land here that may pollute, and they go through wetlands, it gets cleaned up a little bit. We still don't want to pollute, but it helps. But if you live somewhere where they've developed or built on wetlands, you don't have that area to help protect the ocean. So our actions really make a difference, and we can affect it. And we rely on the ocean and the estuary for so many things. Who likes to eat seafood? Yes, I think a lot of people do. Who breathes oxygen every day? I better see everybody's hands, right? So we rely on the bay quite a bit. We get over half the air we breathe from the ocean. All right, any other questions? You've got to be really loud for me. Yes?
Do you eat oysters?
Do I personally? You know, personally, I don't like oysters or other bivalves. They're called bivalves because they have two shells, like mussels, clams. Some people really like them. You know, they're very popular food. Yeah, Miss Mendenhall does. I personally, a little slimy for me. Yeah.
What's the difference between oyster and a clam?
Oh, what's the difference between an oyster and a clam? Good question. Well, oysters are going to hold on to that hard substrate. So they're going to land somewhere and they're going to grow kind of vertically. They're going to grow up, and they grow in big reefs. A clam typically lives in the sand or in the mud, and they have these long siphons, which are like straws, which stick up above, and that's how they filter the water. So slightly different animals, but they're very similar. They both have two shells, and they eat very similar food. Great question. All right, let's see if anyone over here who hasn't asked a question has one. Okay, how about over here? Why are oysters important? Oh, well, who can tell me? Why are oysters important? I mentioned a couple things that oysters do that's really important. Does anyone remember it? Yes. Yes, they're filtering the water, exactly. So that's really important. We need them to filter the water. And if you can remember back to the picture of the tank that I showed you that was full of algae, it was dark, dark green, they can filter that water in less than an hour and make it completely clear. So that's really important. They filter water. What else do they do? Who remembers? They make something. They kind of build something for other animals. Yes. Exactly. They're creating habitat for other animals to live. And we don't need them here as much, but on the East Coast, on the Atlantic Ocean, oysters build really big reefs that actually protect the land, so from erosion. They don't do that in Elkhorn Slough as much, but that's really important if you lived in like the Gulf of Mexico or on the East Coast of the United States. You would need oysters there too. Yes?
I have a question. Did your study make you think of future questions for future scientists?
Yes. My study, so as I explained, when I put those baby oysters out, they were the size of about a pencil eraser. I think a great question to ask would be, what would happen if I put oysters out of all different sizes and compare them? So from the tiniest little oysters that just settled to oysters that were adult size. I think that would be a great question. I also learned that the non-natives in my study didn't have as much of an effect as I thought they would. So I would like to investigate that further. Why didn't they? And my idea, but I would have to test it, is that these oysters live in a place that's really stressful, right? When you live in a place where the tide's going up and down, so you're in the air part of the day, you're underwater part of the day, that's a lot of stress. It's hot part of the day, it's cold part of the day, so it's hard for them to survive. So by having other animals around them, it helps reduce that stress. Oysters would typically live in bunches or groups, right? The oysters I put out were single on the place, so if I put out oyster reefs in groups, maybe it would look different. So yes, I have lots more questions. Yes? Do I study something else? Yes, I've studied lots of different invertebrates. So it's always been invertebrates. I've studied different types of snails when I was living in New Jersey. I had a chance to go down to Baja a couple years ago and study tiny little sea urchins that live in rhodoliths. So a rhodolith is a type of algae that looks like a little tennis ball, and they roll around on the bottom of the ocean. And I spent some time down there diving off an island, scuba diving, and studying these tiny little urchins and rhodoliths that no one had seen before. So I do get a chance to study lots of other things. Yeah.
What inspired you to work with sea creatures?
Oh, and she asked what inspired me to work with sea creatures. It's interesting. I grew up in Pennsylvania where there is no ocean, and I really love the ocean. I always thought about it. I always wanted to visit it. So I would go to New Jersey, and I would see the animals like horseshoe crabs, which we don't have here, and I just loved them. I loved everything about it. So I said, when I go to college, I told my family, I'm going to study marine science, and they didn't even know what that was. They were like, why would you want to do that? I said, it's really interesting to me, and that's what I'm going to study. So things that you find interesting, I would encourage you to pursue them, because that's where you're going to have a lot of fun. If you find something interesting and want to follow that. Yes? Oh, that's so hard. What's my favorite sea creature? That's a really hard question because there are a lot. I would say it's probably a shark. If I had to choose an invertebrate, it would probably be one of the really cool snails with the really detailed shells. I find interesting too. Yeah. I think we're out of time, but you can stay and ask me, okay?
Let's all give our presenter a round of applause.
Thank you. What's your question? Oh, have I found any dangerous animals?